I just want to start by way of a quick introduction. It's a tremendous honor and privilege today to be present at Sina's defense and to be his advisor. Sina came to my lab shortly after he arrived at Caltech, and he looked around and thought it might be a good idea to join. I was a bit flabbergasted. He's a mechanical engineer, and personally, for me, mechanical engineering ends at where I have trouble winding up my mechanical watch. So I didn't know what exactly to make of this, but Sina had a vision for why he thought it made sense to join my lab, and he'll tell you about that today. Sina, there's a term in genetics called epistasis, which means when you have different genes interacting together where the whole is greater than the sum of the parts. And Sina, I think of him, he's like an epistasis generator. He's an accelerant. He makes everything better in science, in engineering, and in life. And he'll tell you today about the science. I mean, it's been an incredible ride. He's written 14 papers, and many of which are already highly cited. And that's his job today. I want to just tell you one or two things about what kind of human being he is. And his humanity is evident in his science and vice versa. While Sina's been here at Caltech, every time he sees a problem, he just tackles it. And he doesn't do it for the headlines or for the acknowledgement. He was an RA here, and there were problems in the student housing, and he went straight to the administration and took a real risk. When COVID hit Caltech, Sina became part of a working group, a very small group with maybe 4 or 5 of us, which met regularly with the provost. And in May, just 2 months after COVID hit, he was arguing for us to have testing at Caltech. And he was arguing against a faculty member who thought the opposite. And he was right. He didn't just talk the talk. When faced with lack of testing at Caltech, he wrote a paper. He surveyed all of the universities in the United States to provide data and evidence of how important testing is, looking at who had testing in place, who didn't, and what the COVID outbreaks were. This was widely cited by newspapers, and it really had an impact well beyond our institution. And he actually developed software to facilitate COVID testing. I hate to say this, especially this week, but it's important to know that a year or two ago, we had a threat on a shooting at Caltech. And Sina was the first student who spoke up to the administration at great risk to himself and pushed back on the lack of response by our campus. I think he literally has made our campus safer for the future. Sina, it doesn't have limits anywhere. When there was a huge blast in Beirut, you may remember a couple of years ago, he sat down that morning and used the G.I. Taylor approximation to calculate the size of the blast. He was the first person in the world to do it. And it had a huge impact because it was cited by newscasts around the world. He is the first estimate of how big of a deal that explosion was. He estimated the blast at 3 kilotons. And last year, just at the end of 2021, the final number came through. It was 2.7 kilotons. It's like amazing, right? And so, in every respect of life, Sina just tries to make the world a better place, and you'll see that comes through in his science as well. So for me to be his advisor was a challenge. I just had to sit down and listen, and that's what I've tried to do: listen and learn and become a better person. Thank you, Sina, and I look forward to your presentation.
Well, I got to say, I was not expecting such a moving introduction, and Leo, thank you. It's been a ton of fun. So thank you all for showing up. I'm really excited to tell you about my work today. So when I came to Caltech, this was in 2017, I did whatever any grad student does. Like I go to labs, talk with other grad students, try to get an idea of what kind of science is going on at this university. And I met 2 really impressive people, Lin Yi and Jace Gehring. And they were both graduate students at the time in Leo Proctor's lab. And it was during one of these discussion sessions that I was shown this video that you're seeing right here. And Lynn and Jace were going on and on about how this video, it epitomizes the types of biological experiments that are yet to come, which could be deployed to better human health. And I sat down looking at this video, like I'm a mechanical engineer, like I see, looks like some kind of fluid flow, maybe there's like droplets being generated, it looks like there's particulate matter, and I was a little bit confused, like you're telling me this thing is going to solve a biological problem. And just to give you guys a little bit of numbers, like in a sense of what's going on here, this is an image of a microfluidic chip. So you can think of like really tiny plumbing. And the size of these droplets that are being pinched off are about 100 microns, so about 2 widths of a very thick human hair. And I learned after talking with Jace, after talking with Lynn, after working with members in the lab and doing a lot of reading, that what's going on here is that cells, which are these really tiny, super tiny dots, which are coming along the bottom, are getting encapsulated inside of a droplet, along with this spherical object, which I'm going to call a bead, to create an isolation chamber for chemical reactions to take place. And this, this is actually really important. And I was sitting down there during my first year thinking, wow, if we can create individual isolation chambers for cells, and we can do all kinds of really interesting chemistry and biology, which I didn't know how to do, but which Jason and Lynn were telling me were possible, this seems like it could be really impactful. And so, I did what any good scientist does. I looked around, I started reading papers, and I realized that the scale of experimentation is exponentially increasing. So what you're looking at here, so each dot here is a paper, and I'm plotting, or it's being plotted, this associated number of cells that are being studied in that paper. And so this is even a little bit outdated. This is from 2018, and it has since increased. So I looked at this and I just got very excited prior to joining Lior's lab. And Jason Lynn being a medical doctor, she's now in med school, they would always talk about the potential health impacts. And we've actually started to see some of the health impacts of this tool, which is called single-cell RNA sequencing. And I'll explain exactly what that means in a bit. But it's actually been shown to be useful as a cancer diagnostic tool. There are papers that show that you could use this technology to curate biomarkers to detect lung cancer. You could use it to identify the effects of treatment on brain cancers. You could evaluate the impacts of therapeutics on breast cancers. And you can understand chemotherapy resistance in ovarian cancer. And so all of these potential health impacts of this technology were just really incredible. But as I kept on learning and reading, I came across 2 mysterious black and white boxes over and over. The papers would always say, we use this on the left-hand side, 10x Genomics Chromium controller to encapsulate cells and barcode the RNA. And the paper would then go on to say, we use an Illumina sequencing machine to sequence the library. And I thought, okay, well, I should just read this. Let me go take the manual from each of these machines and just read about it. So I remember one weekend in Pasadena in Intelligentsia coffee shop, I sat down with Jay Scaring. I pulled up the manual for the machine on the left. It's like 50 pages long. And of course, it's filled with all kinds of chemistry and reactions. I just really had no clue. And Jace asked me, Sina, do you know what you're reading? And I was like, honestly, no, can you help me? So he sat down and he took the time to explain what's going on. So after learning a bit more, I found out this machine on the left, which costs, at the time it was about $100,000. Now it's cost about $50,000. This machine is used to perform what I just showed you on the first slide, this encapsulation procedure. This machine on the right, which costs $128,000, is used to sequence the RNA that's captured. And I'll explain a bit more in a little bit exactly how that works. So this machine on the left, as I mentioned, it operates based off microfluidics. You have cells and these beads coming together inside of an isolation chamber. Now, you may wonder, what exactly are these beads? They seem kind of special, and you'd be right. If you've ever, as a kid, played with these kind of squishy, these hydrogel beads, they're like the sensory beads. You stick them in water and they grow larger and they grow smaller. They're actually mostly water, and you can actually do really interesting labeling on the bead to attach synthetic DNA barcodes that can grab the RNA in the cell. And then once you've done that, you've taken RNA that's been generated from a cell's DNA, and then you've tagged them with a unique barcode that corresponds to each cell, and then with a unique barcode that corresponds to each molecule. You take the collection of these molecules, you put it inside of a tube, and then you sequence it. And then you send it to that machine right there. And then you get back literally 2 text files, or maybe 3 or 4. But it's the text files where each entry in the text file corresponds to a sequence of nucleotides, so A's, T's, G's, and C's, if you remember from high school biology, that from molecules that were actually in the cells that you captured. So what does this mean in the context of human health and the whole procedure of performing single-cell RNA-seq? Well, there are many distinct engineering challenges. First of all, humans are very complex. They're composed of 37.9 trillion cells, and they have different kinds of morphologies. For example, cells that are in your nervous tissue, for example, in your brain, or neurons, are quite different from cells from muscle tissue cells. And you can imagine that there's an interesting challenge of resecting cells from each of these regions in such a way that preserves the state of those cells for further analysis. And so that's one engineering challenge. The second engineering challenge is to take that resected set of cells and dissociate them or break them apart. So that way you can actually perform this isolation procedure to then tag their RNA. That's another engineering challenge. But once you've done that, you can then take your really expensive isolation machine, and you can imagine it's an engineering challenge to figure out how to get cells and beads and some kind of reagents into an individual droplet so that you have one cell and one bead inside of one droplet. Because if you had two beads, that could be problematic for downstream analysis. If you had no bead, you're not going to get any RNA from that cell. So it's a problem getting stuff in, it's a problem getting stuff out. And then once you have that library, there's a really fascinating engineering challenge associated with sequencing the molecules. And this machine right here that I'm showing you, you can think of it as a really fancy microscope. And the way that it works is you have this thing called a flow cell, which is this device right here, this rectangle. And patterned into this flow cell are a set of wells. Now, the molecules that you get from your sequencing library attach to each one of these wells. And if you remember, they're just made of A's, T's, G's, and C's. Then the machine flows molecules to bind complementarily to the double-stranded DNA. And it just takes pictures. So lights up, takes picture, lights up, takes picture. And it builds the molecule from scratch, taking pictures. That's what I'm demonstrating here in this photo right here. It's kind of like a lawn of molecules. And so once you have this stack of pictures, you can imagine there's very interesting image processing. There's very interesting control theory to actually manage the flow of all these things to then generate this text file for all the molecules that were in your cells. And then once you have this text file, and mind you, this text file can be very, very large. I'll show how large in just a second. It's another engineering challenge to take that text file and transform it into a format that's actually usable for biological discovery. And so just to give you all a sense of just some numbers, you can imagine a typical experiment would take cells from, this is sort of a technical experiment, but it mixes cells from human kidney and mouse fibroblasts. It then captures molecules for each cell. You then get a text file that's comprised of 721 million lines, which, compressed is about 56 gigabytes on your computer. And ultimately, you want to transform it into a matrix that's cells by genes. So for each cell, what is the number of molecules that cell is producing for that gene? And so my thesis solves key engineering challenges in single-cell RNA-seq in two main areas. The first area is in cell isolation and collection. So I'll talk a bit later today about how we can perform isolation at scale and how we can possibly perform long time series RNA sequencing experiments at scale as well. The second part of my thesis discusses how to transform this file right here, this really large file, and make it meaningful, transform it into a matrix. So starting with the hardware. So a single cell RNA-seq experiment is limited by cost. So the scale of the experiments are limited by cost. You can imagine this machine right here can process around 10,000 cells per 20 minutes. And that'll cost you about $1,500. And as you saw on the few slides ago, the scale of experiments is increasing, and we're now looking at experiments with millions of cells. So if you want to start spending more and more money to make more and more cells, it becomes really quickly a limitation. So one alternative that you could do is you could actually use open source devices. So takes everything in that black box, exposes it, and makes it accessible so that you can modify things, oftentimes much cheaper. And so an open source pump or open source rig to do this exact thing will cost you around $10,000. But there's still a problem, and that is that one of the components of this open source system is still quite expensive, and that's these syringe pumps. Now, I should clarify that the device above uses pressure-driven flow to get cells and droplets together in this chip. The open source pumps also use pressure-driven flow, but that pressure is imparted by a syringe. Yeah.
Why is there a difference in the cost per run?
So the question was, why is there a difference in the cost per run? I actually think it comes down to proprietary reagents from these companies. I have a breakdown somewhere, I could find it. Yeah. Yeah. So we set out to think about, can we make low-cost syringe pumps that can actually be used in these experiments? And we created a list of specifications. We didn't actually care that the chassis was made of metal versus some other material, and we realized we could actually just 3D print the chassis. We wanted the syringe pump to be adaptable to different sizes of syringes. We wanted to hit the desired flow rate for an open source single cell RNA-seq protocol, namely dropseq. And we also wanted to use stepper motors, Arduinos, Raspberry Pis, devices that can be purchased off the shelf, because we felt that these had sufficient amount of precision that could enable these kinds of experiments. And most importantly, we wanted to be cheap so we could actually make a lot of them. So we built the Poseidon syringe pump system, and it's a set of 3 syringe pumps, although you could print more, which with a stepper motor that drives a screw, which moves a sled, which slides on linear rails, and then as the motor turns, the sled is advanced or retracted, moving the plunger of the syringe, which then pushes fluid into the microfluidic chip. Now, this device is made of 3D printed plastic. To print everything here, it takes about 16 hours. It takes about an hour to build, and it costs $310. And we're actually able to get the kinds of flow rates that we would expect for these open source systems. So this felt really promising to us as a tool. And I should add that we have this device is open source, and we've put all of the files and the software to run it on GitHub. And I'll add one more thing. As part of the system is this device over here, which is a Raspberry Pi with a screen to run the pumps, as well as a microscope to image the process being performed on the microfluidic chip. And this was published. So the Poseidon system is modular. And as I mentioned before, we had this constraint where we wanted to use off-the-shelf pumps, but we actually took it a step further, and we only used pumps that we could, parts that we could source from Amazon. So we wanted to be able to get them from Amazon Prime. And so this is the system. You have a device here, which uses a Raspberry Pi with a touchscreen to control the Arduino board. Now, the control scheme, it was a little bit complicated to put together because we actually had to design a serial interface from the USB port on the computer to the USB port on the Arduino. We had to be able to then send pulse width modulated signals to each stepper motor, so that way we could get precise flow. And as you can see here, each of these syringes will then go into a syringe pump and will then produce flow at the end. So we felt pretty confident that we had a device which we think could work. And so we actually went to test it, and we found that Poseidon is actually quite comparable to commercial syringe pumps at a fraction of the cost. So what I'm showing you here on the left-hand side is an image of a microfluidic chip. Now, this microfluidic chip is slightly different than the one I showed you earlier. It doesn't encapsulate cells, it just produces droplets. And those droplets are imaged on the right-hand side. And the goal of this chip is actually to produce droplets that are monodispersed, meaning that the size of the droplet is very uniform. There's no variation in the size. So if it does a good job, if the flow is stable through these channels, then you'll produce a set of droplets that kind of look like this, packing in a kind of hexagonal pattern. So we used the Poseidon syringe pump system, and we tested it against a commercial syringe pump, this $5,000 Harvard apparatus syringe pump. And what I'm showing you on the right-hand side is the results of those tests. So we performed 3 replicates across 30 minutes, where each 3-minute interval, we're sampling a collection of these droplets and evaluating the variance in their size as a proxy for how stable the flow rate is. And what we see is that actually we hit the target size of around 50 microns diameter in droplets. And actually our variance is quite similar with the Poseidon syringe pump to that with the Harvard apparatus. So this was really exciting to us. We were ready to do inexpensive single-cell RNA-seq experiments at scale. But we still had one problem. And that is that time course experiments can be very time consuming. So you can imagine we have this chip here. We can get stuff into it, but to then get stuff out at a flow rate of, like, let's say 20 mils per hour, if you wanted to do like a, let's say, 100 mil experiment, you'd be waiting a few hours for this. And so we realized, okay, we can actually solve this if we just use, this device on the right-hand side, which is called a fraction collector. Now, a fraction collector's job is to do exactly that. It's to take fractions of liquid from any kind of experiment or any kind of device, and then proportion them into tubes. But then we started to look, and we realized that actually these are pretty expensive as well. Like the cheapest one we found was about $1,700. And we felt we could do better. So myself, along with Ann Kim, a collaborator, another graduate student in the lab, Ann Kill, sorry, we produced the Colosseum fraction collector. Now, this fraction collector, it comprises a few parts. One is a drum with holes in the top. Now, the holes in the top are intended so that you could place tubes to collect fractions from your experiments. There's a dispensing arm, which takes tubing from the output of your experiment and routes it into the tubes. There's a base. There are these two sort of gears right here, which mesh together with a timing belt pulley. And the reason for that is because our device uses only one motor. And that's how we were able to get the cost of this device so low. And we take advantage of this really cool mechanism called the cam follower mechanism, where the dispensing arm has a bearing that slides in a slot in the base of the drum. So as the drum turns, the arm moves in concert. So that way we only need to turn the drum, we don't need to turn the arm. And this was a significant advantage over commercially available systems which had two motors. I should also add that two motors makes the control scheme for managing them very complex. So we could 3D print this device. The print time of the version 1 took 73 hours, but Anne has been working amazing. She's done a really great job on improving this, and we've got this down to around 22 hours. It takes around an hour to build, and it costs $67. So if you remember, the commercial one costs about $1,200. So, and again, this device is also open source, and you can build it yourself, and it's available on GitHub. So we then set out to characterize the device. We wanted to know how good are we at collecting fractions. Now, it seems like kind of hard thing to do because you're literally just getting liquid and putting it into a container. So I won't go through, the extensive characterization that we did, but I'll point you to panel D, where what we evaluated is over a range of flow rates, how accurate are we at getting the amount of fluid that we expect in that tube at that time. And you can see that we're within plus or minus 5%. We could probably do better. And I think there are ways to improve the device to make it better. For example, changing the nozzle so that it doesn't drip when it passes in between tubes, which was one of the biggest sources of error. So this was really exciting. And we put out this paper on Hardware X. So we felt that now we could do time course, open source, single cell RNA-seq experiments at scale and for very low cost. So we were ready to actually start doing experiments. But then the tragedy of the COVID-19 pandemic unfolded and experiments were on pause. And just for those of you who don't know, this is an image taken in our old lab space in Kirchhoff with very beautiful soft white lighting and nice archways. So it took some time for us to get used to Zoom, but I think we sort of got it, even after playing around with it for a little bit. But during that time, I was able to engage in some really exciting computational work. Now, I don't have time to walk you through everything, but I will talk about the results from two papers, mainly this paper on processing single-cell RNA-seq data, making it usable. This was published in April 2021 in Nature Biotechnology, and then the application of that tool to the analysis of biological data to study the primary motor cortex of the mouse brain. So this is a region in the mouse brain. And this was published in October in Nature. So I want to take us back to where we talked about this whole bead and cell process. So we have this isolation chamber in green. We have a bead that has a set of nucleotides, so synthetic DNA sequences, which are bound to the bead, which grab the RNA, the polyadenylated RNA in the cell. So you can imagine the cell has a bunch of RNA swimming around. You put it in this isolation chamber, you break it open, it releases all of its RNA, and you try and grab it with this bead. And then the ultimate goal of this whole procedure is to generate a kind of barcode. UMI stands for unique molecular identifier, which tags the specific molecule. So this is a barcode on the molecule. This is a barcode on the cell. And then you have a sequence, which then corresponds to a gene or possibly a set of genes. So this is the whole idea. And I should also add that the isolation chamber doesn't have to be a droplet. It just so happens that in this 10x machine, which I don't have on the slide, which uses microfluidics, it actually is a drop. It could be, for example, a plate. You can deposit cells into individual wells within a plate and then put reagents to capture the RNA and label them. But in this case, we're showing a droplet. So then I started to learn, what does it mean to preprocess or to process single-cell RNA-seq data? And it turns out it's actually pretty complex. So you send a set of molecules to the sequencer, and you get back a kind of these files that I mentioned earlier. And then portions of the sequences in these files correspond to different things. The blue set of sequences correspond to individual cells. So all of the sequences that have the same blue barcode were derived from the same cell. And that's really important when we go to count molecules. The green portion corresponds to molecules. So all of the molecules with the same green portion are actually the same molecule, but just duplicated. And this is a technical artifact, which I won't explain. There's a little bit of technical nature that I won't explain there, but I'm happy to explain after. So then we take the red portion and we align it to some kind of reference. So maybe for those of you who don't know, there's a set of public databases with references that map sequences to their genes. So there's a nomenclature for the genes. And usually those genes have some kind of function which is relevant to maybe human health or organism health. There's another concept which I'll talk about in a bit, known as an isoform. So this is a stretch of sequence contained within a gene that actually is labeled as the gene, but functions slightly differently. And I'll explain exactly what that is. So we can take our sequences, we can align them to a reference, and then we can count and aggregate the set of sequences into this data structure right here, which I'm calling a cell by gene matrix, where each entry in the cell by gene matrix corresponds to the number of molecules we observed in this set of sequencing files that aligns to this gene that were from this specific cell. So this is the process of preprocessing, and it's the process of alignment and molecular counting. So, okay, we wanted to do this, but we realized there's actually a lot of computational engineering challenges. There's many technologies, so the structure of these molecules may differ slightly. There's a rapid change in the versions. These assays can be very complex. So maybe you have you're tagging proteins and not just RNA. Maybe you're tagging chromatin accessible regions and not just RNA. There's a diverse set of software. They're written in multiple languages. There's a lot of data. And importantly, oftentimes there's no papers, standards, or validations. I mean, even the most widely used single cell analysis tool in the field does not have a paper associated with this, making it very challenging for new users coming to the field to be able to understand what is it that they need to do. So I'm showing you here on the right, it's just a GIF of all the different kinds of tools taken from a GitHub repo. And down here, it's kind of how I felt kind of getting into this challenge. So the state of the art in 2018 for taking these sequencing reads, so down here, and producing this kind of matrix, is a tool by the name of CellRanger, which is made by the same company that produced this device. So, okay, I said, well, we'll try CellRanger. Let's go ahead and give it a shot. So we took a data set of about 10,000 cells. It's the same data set I said earlier. It has 700 million reads. To process this data set took 21 hours, required 1.3 terabytes worth of storage, and required 28 gigabytes of RAM. And oftentimes people think about processing in terms of flops or cycles, but I did a very simple back of the envelope calculation and found that the processing requirements for this one processing run on this huge data set used energy which was equivalent to 2.5 gallons of gas. And I think that's actually really important if we want to think about the health of the planet. We actually need to start thinking about the efficiency of our algorithms. So I teamed up with Paul Melstedt. He's a professor at the University of Iceland. And we worked on developing a tool to go from these sequencing reads to results, namely this specific matrix over here. And these two tools are called Callisto Bus and Bus tools. They're command line utilities that take in sequencing reads from a variety of different single cell RNA-seq assays, and then allow you in a very flexible way to do all kinds of stuff with it. Say you wanted to generate this Cal matrix. Well, there's a set of commands that you can run with Bus tools to do that. Say you wanted to process an arbitrary technology. Well, there's a different set of commands. But it's structured, it's rational, and it's thought out in a way that makes adapting this tool to new sequencing technologies very easy. And we actually published it. This is the paper for that. So we built this tool. Now we wanted to go test it. So we took that same data set and we ran it. And we went from 21 hours, so a full day, to 27 minutes, to half an hour. And the storage requirements went from 1.3 terabytes to 15 gigabytes. And the RAM requirements dropped substantially making this something that you can actually perform on a standard laptop, which I think usually has a maximum of about 16 gigabytes worth of RAM. And processing this data set with our new tool actually requires 50 times less gas. So we're being more environmentally conscious. So our tool is actually very fast and memory efficient. So the plot on your left is a plot that shows for 20 different data sets with varying number of sequencing reads on the x-axis, the processing time on the y-axis. So each dot here is a data set. They're colored by the set of tools that are available to perform this procedure. And what you can see, first of all, is that the axes on the x-axis and the y-axis are on a log scale. And so this is actually, pre-processing time seems to be linear in the number of reads. And additionally, what you can find is that we are performing the fastest on a variety of different data sets with respect to the state of the art. Equally important, on the right-hand side, is memory efficiency. So on the x-axis, again, it's the number of reads in the data set. On the y-axis, it's the maximum memory used in gigabytes. And we're in orange, and Cell Ranger, which is the state of the art, is in blue. And what you can find is that actually we're performing, so these two lines, the bottom line is 4 gigabytes of RAM. So that's the requirement for a minimal computer. This line here is at it's at the maximum, which was about 11 gigabytes of RAM, which is the previous slide. And there's been work done in the lab by some of my colleagues to drop these memory requirements even more. So we're fast, we're memory efficient, and we're actually also robust across species and technologies. So this is an extensive benchmark that I performed on these 20 different data sets. And I won't maybe go too much into the technical details. It's just to say that if you see a separation in these two peaks, it's actually a good thing. And we want really tight peaks on the left and really sparse peaks on the right-hand side. And what's really exciting to me is actually this tool that we build, it's not just a one-off tool. It's a mathematical idea that has been encoded in code and is actually extensible to other sequencing technologies. So during the pandemic, as Lior mentioned at the beginning, I worked with some really incredible people at UCLA, Josh Bloom and Valerie Arboleda, as well as Eliezer Eskin, as well as some individuals from a company called Octant, namely Sri Kashuri and Nathan Lovett, as well as Laura Lubert in our lab. And we worked to develop a sequencing-based diagnostic test, which takes sequencing data from patients with COVID-19 and actually produces a diagnostic result. And this assay now, everyone knows it as SwabSeq. It was actually their very first test that was performed at Caltech. It was performed for, I think, a few months. It's still being performed at UCLA. And recently, the University of Arizona is starting to perform this test to detect COVID-19. So I think it's a testament to the modularity of the tool that we can take something that was originally for single-cell RNA-seq and then apply it to something that's completely different. And we also publish this workflow in scientific reports. Okay, great. So we can capture cells, we can do time course experiments, we can create these matrices with efficient and uniform data processing. So now let's actually, as at this point in time, I think like during the pandemic, I got really interested in learning more about biology, namely the brain. I think it's one of the last final frontiers. And I actually teamed up with a consortium called the Brain Initiative Cell Census Network. This is a collaboration funded by the NIH, which was started under the Obama administration, which has put together vast funds to understand the human brain and to understand health and human health and human diseases that result in dysregulation in the brain. So I worked with this collaboration. I worked with this consortium, and they studied not the human brain, not the monkey brain, but the mouse brain, which, as you can see, is really tiny. And we didn't even study the whole mouse brain. We studied a very tiny sliver of the mouse brain, known as the mouse primary motor cortex, which is also shared in monkeys and humans. Now, this region of the brain is really interesting because it's responsible for voluntary motion, so the initiation of voluntary motion. And it was kind of at the time that we published this, and there's an issue in Nature dedicated to multiple papers that study this from various angles. It was the largest data set produced of the mouse primary motor cortex, and we analyzed the RNA-seq data. So I just want to remind everyone that in each cell, you have a process of taking DNA, transcribing it into RNA, and then there's cellular machinery that takes that RNA and recombines portions of it into individual transcripts or isoforms. And it's those isoforms that are then transcribed to produce proteins. This is the central dogma of biology, which is usually taught in high school. And standard single-cell RNA-seq processing tools take data generated from these isoforms and actually produce a matrix which is at the gene level. So just aggregating it at this level. But it's missing out actually on a lot of really interesting biology that happens at the isoform level. You can imagine an instance where maybe a cell is expressing this gene, but there's a variation in the amount of expression between these two isoforms. So what I sought out to do was to take the tools that we've developed and apply them to the mouse brain at the isoform level, a more granular level. So we teamed up with some folks at the Allen Institute, namely Honghui Zhang and Zhizhen Yao, and produced the huge data set. I mean, the amount of data I think is really stunning. We produced 3 different RNA-seq assays, and I'll explain to you what they are. The first one in the middle is so we took the mouse primary motor cortex, and then the first one in the middle is the 10x assay. So that's the original assay I explained to you at the beginning, where you partition cells into individual droplets and sample their RNA. This assay has the benefit of being able to sample a lot of cells, like a really huge amount. But one drawback of this assay is that you're actually only getting sequencing data from one end of the RNA. You're not able to get sequencing data from the rest of the RNA in its current form. And what that means is that if you want to be able to disambiguate sequencing reads that correspond to isoforms versus genes, it becomes very challenging. So in order to combat that challenge, we generated data with an assay called SmartSeq, developed in Richard Sandberg's lab at, I believe he's at the Karolinska Institute. And this assay has the opposite problem. You don't really get many cells, but you get the benefit of being able to sample sequencing reads from across the entirety of the RNA, which allows you to disambiguate these isoforms to be able to get isoform level resolution. The third assay that we used is this assay called MERFISH. Now, this is a spatial RNA sequencing technology developed by Xiaowei Shuang at Harvard. And this allows you to get RNA measurements in situ. So you take a tissue, and instead of having to dissociate all the cells, you just lay it out on a plate, and you use microscopy as a readout for where RNAs are and how much RNAs there are. So with all these 3 data sets, we were able to use these 3 ways of measuring RNA in concert and not in competition. So let me explain what's going on. There's a lot going on. So we took the 10x data set generated by this machine, and we used it to perform cell type identification. So what that means is we took this cell by gene matrix and we grouped cells together that are more similar to each other and then less similar from other groups of cells. Now, this is called clustering in standard machine learning parlance, and we actually give a name to this cluster in order to form a common vocabulary with the biologists who are actually studying these cells. And the name for that cluster is called a cell type. So each one of these circles that you see here is representing a cluster of cells known as a cell type. And what you're seeing in the gene space is the expression or the number of molecules for that cell type for different genes. But with so we use the 10x to create the cell types. But with the SmartSeq data, with the isoform data, we can actually resolve for a single gene which isoforms are actually higher, which ones are lower in that specific cell type. We may not get as many cells, but we're still sampling across the wide range of cell types, getting isoform resolution. And then we can take the spatial data and tether the cell type results that we get spatially. So now we can get cell types here, we can get isoform data here, and then we can map it spatially here to then generate an isoform cell type specific spatially resolved map of the mouse primary motor cortex saying, this cell type is here and is expressed in this specific isoform. And that has not been done before. So as I mentioned before, we take the 10x data and we do cell type identification. Now, before you crucify me, saying I made a UMAP, this is actually an embedding made with a neighborhood component analysis developed by Jeffrey Hinton. And what you're seeing here, each dot is a cell, and I'm painting the cells by the cell type that they belong to. And you can see we have a set of distinct cell types. in the 10x data. We have a set of distinct cell types in the SmartSeq data, which we can get based off a map to the 10x data. That's at the gene level. This is at the isoform level. So much more resolved, but fewer cell types. And this is important because it allows us to understand when there are hidden isoform changes. So when there are changes, for example, at the isoform level, so that's these two violin plots. So each column is a cell type. And then the bottom row is the expression of an isoform of this gene, OXR1. We find that actually there's an isoform shift between these two cell types when there isn't a shift at the gene level. Now, I don't have time to explain exactly how this works or the complexities of it, but it's just to say that by looking at the isoform level, we can get expression differences between cell types that could possibly be clinically actionable. We can also get spatially variable gene expression with the Murphish data. So on the left-hand side, what I'm plotting, each dot is a cell, and the x-axis is microns, and the y-axis is also in microns. And I'm plotting in black the identity of that cell with respect to its cell type, which is labeled here. I'm computing the centroid of that cluster, and then asking how far away is that cell type from the boundary of the cortex. So if you think of this as the mouse brain, we sliced it, we took a small section, and I'm asking how far from the edge of the mouse brain is this cell type. And then I look along the depth of the cortex to see if there are any spatially variable genes. You can imagine that maybe this is the case, maybe it's not. We didn't find really any that we felt confident calling as spatially variable, but we believe that further research in this area is warranted. So one of the main results from this paper is that we can get a spatially resolved isoform atlas of the mouse primary motor cortex. Now, just to give you some context, this is the mouse brain right here. This is a sort of cross section on the side. This is a cross section front to back. And actually this little sliver pointed to by this arrow is what we're studying. That's the mouse primary motor cortex. We are painting here, this is the spatial RNA data, where each dot is a cell, we're painting it by its cell type. On the right-hand side, we have the cell type identification. We have the cell type, spatial cell types in this column here, and we're painting them by which, where it actually is. Each row corresponds to a cell type. Each column corresponds to a marker gene that was found with the spatial assay. But then we're doing this inference approach where we take the isoform data and saying, okay, that we know that cell type actually is marked by this specific isoform. We know that cell type is actually here in the RNA data, in the spatial data, so we can infer that this isoform is actually what's being expressed in that cell type in that location. And so the violin plot is demonstrating the expression of that gene in that cell type, and the number corresponds to the specific isoform of that gene that is upregulated in the isoform data. So this was this is sort of kind of the main result, and what you would expect to see is this sort of patterning along the diagonal here, where there is a sort of one, maybe two genes or isoforms that really mark that cell type. So this is really exciting to me because, you know, lower costs of sequencing. So maybe I'll just explain what this plot is first. So on the x-axis is the date, and on the y-axis is the amount of data on the SRA. This is a public sequencing database. And each dot in red, each dot in this kind of greenish black is the amount of data on these public sequencing databases. Each dot in red is the cost to sequence a human genome. So this is kind of a metric that most people go by. And what you see is that as the cost to sequence a human genome is decreasing really drastically, the amount of data is increasing. So lower cost means more data and hopefully more discoveries. I believe that biological discovery is actually extremely challenging. On the left, we have this mouse primary motor cortex. So we just looked at a sliver of it. I mean, mouse is 36 times smaller than I am, and we have so much data, and we still we don't know much. Now I'm going to quote a former Caltech professor, Ray Deshay here. He has, we really know how to take this rover, size of an SUV, a mini SUV, take it from Earth, put it in a rocket ship, take it to another planet, land it down within feet of where we think it's going to go, and then send a helicopter up to take pictures of that device, of that rover. And we can do that like with incredible precision. We can compute that, we can calculate that. It's extremely challenging to calculate in the same way on biological data when we're trying to both learn new biological discoveries, as well as develop the tools to learn those discoveries at the same time. So it's actually not rocket science, it's biological science, which means I actually need to change my tie. This seems kind of inappropriate, but it's a good thing that I came prepared because I have another tie right here. Let me just go ahead and put it on. So, sorry everyone on Zoom. All right, let's see if I can do this under pressure. 1, 2. Okay. I think it looks pretty good. What do you guys think?
Oh, wow.
So I'll just add, this tie is quite special because it's a replica of a tie designed in 1955 by a man by the name of George Gamow, who was a physicist, who teamed up with a few professors, some of whom were at Caltech, namely James Watson, Max Delbruck, Richard Feynman, a few professors, one from MIT, Salvador Luria. And these individuals got together, I'm trying to fix one. These individuals got together in order to study how RNA makes proteins. They really wanted to know. So they got together and they created the RNA tie club. And this was the design that George Gamow proposed. So I made a replica because really it's not rocket science, it's biological science. So I just want to thank, I think acknowledgements and thank you was extremely challenging because you often, you forget someone, or, but I just got to say, like, this group of people right here, it's about like 15, I think. They're really special. They've really made Caltech a very special place for me. They've helped me in numerous ways, scientifically and non-scientifically. They've supported me. And I think really there's a huge amount of concentrated knowledge here that I believe is currently being tapped to really change genomics and single cell analysis for the better. And because I think biological science is one of the most challenging things we can do as a species, studying ourselves, I believe this diverse team of individuals can tackle that challenge and will. And I believe that that's extremely important. And I'll just end on one more note. In all these data sets that I've analyzed, all the data sets that's being produced, all the data sets that are on these databases, it's the result of a loss of life for a person, for an organism, and we should not forget that. When we're doing data analysis, when we're putting up these really beautiful images, when we're getting awards from the papers that we make and the citations that we get, we should not forget that it's the organisms that have sacrificed their lives, that we've treated with various conditions and drugs to gain those discoveries. So I really hope, like, in the next, however long we're doing this, that we really pay it forward. I think that's important. Thank you.